alpha 2b3 activation, alpha granule secrase, and 10 screws. You can see here that with in the there's a reduced alpha 2b3 activation as well as P-selectin and ATP secretion in the platelet specific PKM2 definitely compared to the wild type control. Then we look at the clot retraction, which is this is a assay to understand the integrin outside insulin signaling, because then fibrinogen binds to integrin and leads to outside insulin signaling. So these are the wild type platelets. These are different time points, and this is the platelet specific PKM2 deficient mice. And you can see that the clot retraction was inhibited in the murine platelets lacking PKM2 after thrombin stimulation. And again, this is the same mechanism what you show in the mouse plate. With the human platelets, we want to confine the mouse platelets because these are specific mouse which are deficient platelets. You can see here that there is a decrease in the phosphorus AKT, as well as there is a increase in the PSK3 beta by total G squared beta, as you can see here. So this is all good. So this is all in vitro. So the question is, can we reproduce this in vivo? So in my lab, we do a lot of intravital imaging, which is real-time imaging. So in this assay, can you see this movie? Yes. Okay. So here is a procedure where you level the platelets with calcium green and you infuse in the retroorbital plexus, and then you make an injury on the artery using a ferric chloride, and then you can see the thrombus formation in real time, and then you can record, and then you can evaluate all the videos. So what you saw here, this is initially you saw there was some thrombus formation, and this is 18 minutes after 5% ferric chloride injury. So very powerful technique. If somebody wants to test a drug, you can give the mouse. If the drug interacts directly with the platelet receptors and inhibit, you can see the assay in 1 hour. It gives a direct evidence whether the drug is working, is inhibits platelet or not. You can have all this effect, but here you can show the in vivo effect in a real in vivo model. You can also test other drugs. You can give the mouse, you can take out the platelets and then see the effect, but this is a direct evidence, and you can record everything. But these are carotid arteries, because my lab is interested in stroke, and most of plaques get into carotid artery and it goes up. So this is a model. But these arteries are very big, and it's not easy to image. So we have another assay, which is a microvessels. These are like 150 micrometer vessels. This is a vein, this is artery. You can see a nice vessel. There's no thrombus formation. So this is a baseline. And these are single platelet adhesion, which is after 2 minutes after ferric chloride injury. Now, because what this does is this ferric chloride injures the endothelium, exposes the subendothelium, then collagen exposed, and the platelets start binding to the collagen receptors. This is 9 minutes after injury. So if there is a drug or if the molecule is playing a role, you can see inhibition in this thing. So this is just 9 minutes after. You can see a nice thrombus formation on the injured vessel. And this vessel is occluded. So you can this is after, I think it's after around 15 minutes, and you can see. So by this technique, using microvessels, you can quantify single platelet adhesion, which is the first step of thrombus formation. You can look at the emboli, you can look at the embolization, you can look at the mean occlusion time. So you can use a lot of parameters. So we use this assay, and then what we did is that we use a platelet-specific knockout mice. On the left-hand side is shown the microphotographs, and this is shown the quantification. And this is the thrombus growth. So what we found that if the mouse which has deficient of PKM2 in the platelets, the time to occlusion was prolonged, as well as the thrombus growth fold was decreased in the platelet-specific PKM2 deficient mouse. And similar results were obtained with the female mice. So these were all with knockout. Then we wanted to look whether, because we are affecting the thrombosis, so the important question is whether it also affects hemostasis. And if the drug affects hemostasis, then it's like other drugs. So why people are interested in alternate drug? So as you can see here, that the bleeding time was not different, but they were compared between the wild type mouse and the platelet-specific PKM2 deficient mouse, both in male and female. So now we want to test the efficacy of the drug. So we use the same wild type mouse, and we use 2 different concentrations, and we have some people who are like MTech in pharmacology and they came from industry, so they calculate the dose which mouse can go. So you give oral gavas, and then you can see the time of occlusion. This is the control with the vehicle control, and these are two different concentrations. You can see a very good effect. This effect is better than the knockout mice. And the reason is, I'm sure somebody is going to ask the question, but I can give the answer. The reason is because there we are using a plater-specific knockout. Here we are giving an inhibitor which also affects the function neutrophils and monocytes, which also contribute to thrombosis. I'd like to conclude my talk. Hopefully, what I've convinced you is that platelet activation increases PKM2 dimer formation, which regulates PI3 kinase mediated AKT and GSK3 signaling, and thereby modulates platelet function arterial thrombosis. Targeting dimeric PKM2 inhibits arterial thrombosis without altering hemostasis in vivo. So what I showed you is all healthy donors, but healthy human does not get disease. So right now what we are doing is, we are doing some studies where we are trying to target dimeric PKM2 and looking at whether it affects platelet function in the platelets from the coronary artery disease patients or the obese patients. We are recruiting some donors and controls. It's very difficult to have diet-induced obese patients and they match the same control with the same ethnicity, with the same age. So we are doing and working with some doctors. I have some couple of MD PhDs in my lab, so they are working on that. And this is a new area we are targeting. So what is the effect of targeting PKM2 dimeric on metabolic pathway platelets? And looking at the metabolic data, we have found multiple targets. And that's the whole area of, you will see in a couple of years, a couple of papers coming from my lab, and nobody is working on this area. This is a very hot area, and this was by accident. This is all published in Blood, and we have filed a patent, and we are looking for that. So, this is my lab. I used to have like 10, 12 people. Now this is a lab people. Most of the work, what I showed you, is done by Manas, and the other people who left my lab have become research professors in different universities. And my lab is funded from NIH R35, NIH R01 from NINDS, and the NIH SPAN, where we do multiple testing. And I am also the actual investigator of the American Heart Association. And recently, I was given a Distinguished Scholar Award and funds for developing this into a novel therapy. Thank you.
Yes, thank you. Thank you, Dr. Chauhan, for a very excellent presentation and very basic research, yet very important and had a lot of translation. How PKM2 inhibits platelet aggregation. It's ML265, I think. You know, I remember when we were in Leh, we used to have a lot of venous thrombosis patient, and they used to die within few days and without, clinician diagnosing it, it is because of the venous thrombosis. So we took a very big project analyzing it, and mainly the clotting factors were responsible. Some of them were getting activated, which was leading to platelet aggregation. So, very important study. It had a lot of clinical implications. Any quick question, one or two?
So before somebody asks a question, so I will tell because.
Good morning, sir. I'm also working on the same area. As you show the plot of dimer and tetramer, as we know, the dimer and monomer also, they have nuclear translocations. So it is better to check the monomer also.
Yes, but these are different type of gels, not it's a non-native gel.
Sir. If you should run a native gel also, we get the 3 bands, monomer, dimer, tetramer, in the same level. So it's better to check the monomer also, because monomer and dimer, they have the nuclear translocation function. And one question, if you knock out the PKM2, there is a feedback loop. So it's better to check the PKM1 level as well.
Yeah, we have checked. There's no difference. But you can follow the papers, you will find all the data there.
Okay.
So because in half an hour I cannot come, so you can. So we have all type of mouse. We have neutrophil specific knockout PKM2. We have a myeloid specific knockout of PKM2. We have the endothelial specific PKM2. We have the smooth muscle specific PKM2. So we have multiple projects. We work on multiple things. You can find all the data. You follow my papers. Some have been published.
You use one activator, one DASA is also there. It's better to check the another activator. And if you check the inhibitor also, it will it will increase the research area. Like you test the activator, you test whatever the effect of inhibitor as well on that simultaneously. So it will be more clear picture.
You are right. So data.
He will be available during lunch. You can discuss. Maybe we can have one or two more
Okay,
questions from
oh,
others.
thank you,
Yeah.
sir.
So I just like to comment on what she said. So I was several years back. I think they had a paper from Lay about DVT, and I think it was published in Blood. I have not shown the data on DVT, but we have a very. So one of my junior faculty got a grant. We have data on DVT. It works very well.
Thank you. Dr. Chan, just one simple question actually. Yeah, what about like, I think this PAR antagonists like ergotroban and all, actually they are available for the similar kind of indication. Whether like this PKM2 has any effect on the PAR pathway also, protease activity receptors pathway?
Yes, I showed the data on the thumbnail.
So is going to inhibit actually that molecule actually, what you make a PKM2 molecule?
So that this inhibits the pathway. So only the point is here. So if you see that data, there's no bleeding.
Okay,
If you use a high concentration of any agonist, this is not going to work.
okay,
If you use suboptimal concentration, it's going to work. And that's what we do with aspirin and other things, right?
okay.
So if you use the aspirin and if you use a high concentration of this thing, aspirin doesn't work. But if you, that's what we want. We want to inhibit the hyperactivation of platelets so that person does not die. But if you inhibit completely platelets, so it's like putting more gas, so in US we say gas, so putting
Okay.
fast accelerator. You can go at faster rate, right?
Okay.
So if you want to go from here to there, right? So if you put more gas, so you are faster going there. But if you put less gas, you will still reach, but more time. That's what we want. That you inhibit hyperactivation of platelets, which affects in the type into obesity or other conditions, and then prevent platelet and make platelets calm. Then inhibiting the whole pathway, because same pathway controls thrombosis and the same pathway controls hemostasis. So there's a fine balance.
Okay.
So if you repeat the experiment with a high dose, it's no more.
Okay, thank you very much. Thank you very much for your talk. Now I would like to invite Dr. Surajit actually Karmakar from INST Mohali for his talk. Actually, he is going to talk in the topic which is the preclinical therapeutic implication of nano melatonin for neurodegeneration and inflammatory disease. He is one of our collaborators also. Now, Dr. Surajit, please.
Good morning. Thank you for the invitation and the kind introduction. So I am Surajit from the Institute of Nanoscience and Technology. So today I'm going to talk on preclinical therapeutic implication of nanomelatonin for neurodegenerative and inflammatory disease. Basically, last few years we are working with this molecule, melatonin, which is neurohormone and released from the pineal gland and has control over the sleep-wake cycle and the circadian rhythm. So basically, the melatonin synthesis and release is controlled by the suprachiasmatic nucleus in the brain. Suprachiasmatic nucleus in the brain, and when light falls on the retina, so it got stimulated and causes the inhibition of the synthesis and release from the pineal gland. So, but when the light got diminished...
Surajit, can you use please mic actually, because, thank you.
So when the light got diminished during the late evening, so then the suprachiasmatic, that inhibition removed, and it started the release, synthesis and release of the melatonin, and it reaches in the peak during the midnight, and that introduced the sleep. So this is their biological function. So basically, the mechanism of action of melatonin is there are melatonin receptor, MT1 and MT2. They are G protein coupled receptor. So when melatonin as a ligand bind to it, it causes the inhibition of the adenylate cyclase as well as the guanylate cyclase, and to reduce the cyclic AMP and cyclic GMP. On the contrary, they stimulate the phospholipase C to trigger the IP3 and diacylglycerol mediated pathway for the activation of the downstream signaling. And there is also receptor present in the retinoic orphan receptor in the nucleus, and also in the MT3 receptor available in the cytosol. The receptor independently it can execute its action. So when there is a inhibition or absence of melatonin, it causes the sleep disturbance, and it is also associated with several kind of diseases, life-threatening diseases. The major gateway is the inflammation, and that leads to these several life-threatening diseases like cancer, diabetes, cardiovascular, neurological. So on the contrary, melatonin having the well-known anti-inflammatory, antioxidant property that is executed mostly due to the inhibition of the NLRP3 inflammasome, the caspase activation, and the cytokine release and the pyroptosis. It has also the inhibitory action on the nuclear factor kappa B to translocate to the nucleus to prevent the COX and INOS mediated pathway inhibition. So melatonin is very well known as a supplement or as a over-the-counter medicine to use to correct the different kind of insomnia, and it is well known reported in the literature having these beneficial effects that like immunomodulation, regulation of the circadian rhythm, mitochondrial function, so anti-inflammatory, antioxidant property. But when we try to use this melatonin as a molecule exogenously, it faces certain challenges. So those limitations are they are having very short half-life and the poor solubility, low viability, extensive fast pass metabolism they are subjected to. So we hypothesized, if we use a appropriate nanodelivery system, we may overcome these certain challenges and we can potentiate its therapeutic efficacy. So with that aim, so we have prepared different kind of nanoformulation in our lab utilizing this melatonin. But today I'm going to talk with the, melatonin-loaded polydopamine nanoparticle or nanocomposite that we have prepared, and we have explored this against this Parkinson's, this Alzheimer, and the diabetic retinopathy. And these are all preclinical models. So first, I'll touch the three. So I'm going to, go very quickly. So the first project is the, exploration of the melatonin polydopamine nanoformulation for the Parkinson's disease therapy. So we have prepared and tested whether this, nanoformulation having some therapeutic efficacy against the Parkinson's disease. So I'm just skipping the, introduction because we are out of time. So basically, Parkinson is a dopaminergic neuron death, and there is a cytoplasmic, like Lewy body, deposition. So those are all characteristics. But we have particularly focused here synucleinopathy. Synucleinopathy, what is that? So there is a, like the alpha-synuclein get highly phosphorylated at its, serine 129, and that causes deposition of the as a Lewy body, and that is toxic and causes the neuronal cell death. So we targeted them. So, and why this combination? So as because there is dopaminergic cell loss, so we thought maybe if we can use this dopamine, that may replenish those kind of dopaminergic, neuronal needed. And melatonin is already known for the, cytoprotective. So this combination, that's why we have tried here. So this is the nanoformulation we have prepared. So utilizing the modified reverse emulsification technique, they are showing 70 to 90 nanometer with a negative zeta potential. So utilizing them, first we have developed our in vitro PD model. So we have used the rotenone, that is a very well known, like the PD model introducer. So we have used two neuronal cell line, SHSY5I and IMR32, and we have checked the cell viability, and we could find the IC50 value. And further, we have propagated all the work with the SHSY5I for this project. So after determination of the IC50 value, we have challenged the cells with this IC50 value, and then we have treated them with this nanoformulation to see whether they have protection or not. So what we could find here, so they are rotenone causing about 500 nanomolar, causing like 61% of the cell death. So when we use the cytoprotection getting into the means like the cell viability is getting up to the 92%. So significantly prevent the rotenone-induced cell death. So reactive oxygen species generation is a rotenone-induced model. it's a common factor. So we could see the treatment, the nanoformulation treatment, significantly reducing the ROS level and preventing the apoptosis that induced by the rotenone here. Then we have checked the mitochondrial membrane potential. So you could see the utilizing the JC1 dye. So the monomer is the, like the damaged population. You can see the green one here. After the rotenone, it is, lightened up. So that means they are introducing the mitochondrial damage, which is prevented following the this nanotherapy. And the last column, if you can see. Also, there is a upregulation of the caspase-3, you can see after the rotenone treatment, that also inhibited following the therapy. So they are working through this pathway. Then with the RT-PCR, we have checked certain, anti-inflammatory genes and the apoptotic genes. So we could see the rotenone-induced, rotenone is causing like the interleukin 1B, NF-kappa B, AKT, so they are highly, upregulating. So that is, checked or inhibited following this therapy. And as because we said like that we are particularly targeting this alpha-synuclein, the synucleinopathy. So monomer to oligomer is a problem. So for the Lewy body formation, so you can see after the rotenone treatment, the last column of the Western blot, there is a oligomer formation, the trailing. So that is inhibited following the dose-dependent treatment. If you see the left column. And also there is inhibition of the caspase-3. Then organotopic brain culture model, we could see the rotenone is, causing the alpha-phosphosynuclein here. Also the activation of the caspase-3. So that is diminished or inhibited following the nanotherapy. Interestingly, rotenone is causing the, we also check the tyrosine hydroxylase. So in the rotenone panel, you can see that is, completely abolished there. So that is retained in the basal level following the nanotherapy. So they are also protecting the dopaminergic neuron in the brain slice culture. Then we have checked the biodistribution. They are well biodistributed and excreted from the system time dependently. And we could see they are located in the brain for a longer period of time. And after the H&E staining, we could not see any kind of aberration in the histological profile. So they are highly biocompatible. Then we have developed the rotenone-induced PD model, and the gait analysis we could see there there is a significant behavioral changes and the sensory motor dysfunction that triggered by the rotenone is prevented by this nanotherapy. And this is the model after the establishment. You can see the dopaminergic neuron is completely lost in the PD model. So, which was retained, after the treatment here in the basal level, and also the alpha-synuclein that is, gone up through the rotenone treatment, also gone down in the treatment group. So we have represented graphically that one. They are significantly doing this. So we can conclude this nanoformulation is causing the inflammation, anti-inflammatory as well as the anti-apoptotic effect, and reducing the serine phosphorylation at 129 to execute this therapeutic efficacy in this preclinical PD model. So next, we have used the same nanoformulation, but it is differently prepared. But the key goal is here to check whether they have the anti-aggregation property against the beta amyloid. So this is the pathway. You probably know, when the amyloid precursor protein produce this amyloid beta short peptide, so that form the oligomers, and then they get aggregated and form a, cause of dementia and the amyloid plaque in the cells. So we targeted and try to see which pathway or which steps this therapy can, disintegrate or prevent the aggregation in our model. So first we have checked the stability and in a different pH. So we could see they are highly stable, but they are, degrade in the higher pH, means the alkaline condition. They are highly stable in the NIR, charge, but they are cannot be sustained into the H2O2, And after that, we have checked the dose dependently the photothermal ability of this nanoformulation, shining the NIR laser. So we could see the dose dependently, when we concentration dependently use them, so they generate the higher temperature. So, and from there, we have done this experiment where we have intermittently shined the NIR and turn it off. We could see the temperature is rising when we are turning on the NIR light, and it is going down also when we are turning off. So this is the NIR image of the particle. And also we could see, as because this is also the melatonin encapsulated formulation, so their releasing profile is they are releasing about like 52% when we are not shining the NIR light. But when we shine some point the NIR light, they are releasing the burst release of the melatonin in this system. So we have done, as because our goal is to check whether they have the anti-aggregation against the beta amyloid or not. So we have done two assays. One is the amyloid beta aggregation assay, and another one is the amyloid beta protein misfolding cyclic amplification assay, the lower panel, the first one. So and we have monitored their aggregation profile with the type 11T. So what we could see in the both cases, they are preventing when we introduce the nanoformulation to the reaction mixture, they are preventing the growth phase. And when we further charge with or shining with the NIR light in the third panel, they are completely disintegrating those aggregation. So TMMH representation here to see that kind of disintegration property. The third one is the NIR. So they can, this nanoformulation can able to quench the ROS. We have done that one. And their axonal growth assay we have tested. So they are highly, biocompatible till 15 days we have observed. So there is no toxicity. But when we introduce the amyloid beta to this axonal growth assay, they immediately die within the 96 hours. So this is another in vitro assay where we have used the amyloid beta to generate the AD model. So you can see after the treatment with the amyloid beta, the plaques are available. You can see within the cytosol, the red fluorescence is representing. So the treatment with this nanoformulation can reduce that amyloid plaque. And when we shine the NIR to it, it is completely inhibited or disintegrate that plaque within the cell. We have also the mouse brain slice culture we have done, where we can see the amyloid beta treatment is depositing that plaque within the mouse brain culture, and there is also activation of the microglia. So that is reduced, and NIR shining completely abrogate them. So also we have done the ocadoic acid model induced AD model. So there we can see that when we treated with the ocadoic acid, amyloid beta is upregulated. And in the second, Western blot, you can see the similar condition when we treat them, that upregulation is downregulated and completely inhibited following the nanoformulation with the NIR. So we can conclude like that they have the anti-aggregation property, this nanoformulation, and the photothermal property and cytoprotection property against this preclinical Alzheimer's disease model. And this is the last project. So where we have used the dopamine melatonin nanoformulation for the diabetic retinopathy. So basically in the diabetic retinopathy is a diabetes mellitus associated complication where the neuronal apoptosis and the microvascular complication happen due to the highly oxidative stress, inflammation, apoptosis, angiogenic factors. So dopamine and melatonin both having anti-oxidative, anti-inflammatory, anti-VEGF activity. So why not we try to develop a therapy? So this is our model. So, and this is our hypothesis. So based upon this property that we have tested against this diabetes retinopathy model. So this is the synthesis and characterization method. So they are spherical inside, and there is a 90 to 120 nanometer in range, and there the FTIR and the Raman spectroscopy showing they are non-covalently interacting with them. So they are becoming crystalline to the amorphous in the XRD study. They are encapsulated in the nanocomposite by the DST study, and they are control release profile we could see.